gram staining, how to do gram staining, what are the steps. But first of all, we'll recap a bit. Okay, what are the different types of staining techniques? Again, you can read and tell me. Come on. Simple staining, negative staining, differential staining, special staining. By the color, can you tell me the dye? What dye did you use in simple staining? Methylene blue. In case of negative staining, India ink. Some of you have drawn India ink in your copy with red color. Okay, India ink should have a black background. Okay, please correct it. In case of differential staining, we'll learn about gram staining today. What was the special stain we learnt about? Again, Albert stain. Right. So today we'll learn about gram staining and its steps. But first of all, we'll need to know about the theory. What are the principles behind the gram staining? What is the cell wall difference between a gram positive bacteria and a gram negative bacteria? Thicker peptidoglycan layer in case of gram positive bacteria, and gram negative bacteria have a thinner peptidoglycan layer. Okay, gram positive bacteria also has strong cross linkages between the peptidoglycan layers. Okay, as well as gram negative bacteria has lipopolysaccharides LPS layer. Okay, in case of this gram staining, the primary stain, that is the crystal violet, we use crystal violet as the primary stain, and then we pour iodine, grams iodine we use. Okay, grams iodine and crystal violet combine to form a crystal violet iodine complex, CVI complex. CVI complex is retained within the cell wall in the cytoplasm of a gram positive bacteria. But in case of a gram negative bacteria, since it has a LPS layer or a lipopolysaccharide layer, don't say LPS in the exam, okay? You have to say lipopolysaccharide layer, which is disrupted by the third step of gram staining, which is decolorization. Okay, first step is the primary stain, second step is the mordant, that is the iodine. Okay, third step is decolorization. We use acetone as the decolorizer, and then in case of gram negative bacteria, we have to know what if gram negative bacteria is present or not. So after decolorization, by decolorization, what do you understand? Primary color will be expelled out. Okay, decolored. Okay? So the negative bacteria will not have any stain at that point. For that reason, we need to use a counter stain or a secondary stain. In our case, we will use safranin. Okay? So safranin will enter the cell wall of the gram negative bacteria and will stain it pink. Crystal violet, in case of gram staining, crystal violet will give you a purple color, not blue. In your copies, the gram staining, some of you have done with blue color. Do not do it with blue color, do it with purple. Okay, and safranin will give you a pink shade. Okay, so this is a bit of a history. It was developed by Hans Christian Gram in 1884. Okay, it is used to differentiate between, you already know, gram positive and gram negative bacteria, and these are the components. First, the primary stain. We use crystal violet here, but methyl violet and gentian violet can also be used. Gram iodine, which acts as a mordant. It is bold here. So what is a mordant? Exactly. Please. Okay. It will fix the stain. Okay. It will fix the primary stain. It will form the crystal violet iodine complex. Okay. So it will fix the stain within the cytoplasm of the bacteria. Okay. And decolorizer, we use acetone, and for counterstain we use safranin or Carbol Fuchsin. Here we will use safranin. So first, the procedure in non-greasy slide, you have to first label it, and then a smear. The smear will be prepared by pouring a drop of saline and taking a scoop with the help of an inoculation loop from the culture media and making a thin oval shaped smear on the at the center of the glass slide. Okay. Now the steps of gram staining. Okay, we'll move on to the steps later. First, gram-positive cocci. Gram-positive cocci, what will be the shape and the color? Spherical purple. Gram-positive cocci. Gram-negative cocci? Pink spherical. Come on, say it with me. Gram-negative bacilli? Rod-shaped and purple. Gram-negative will be pink. And gram-positive bacilli will be rod-shaped and okay. Next, these are the examples. You need to know the examples. Once you do the gram staining, okay, the examiner might ask you, okay, what are some of the organisms that are gram-positive? Suppose you have found a gram-positive bacteria through your gram staining, you like focus it under the microscope and the examiner asks you, okay, you have found a gram-positive cocci. Tell me an example of gram-positive cocci. Then you have to say Staphylococcus aureus, Staphylococcus pyogenes. These are the examples you have to give them, okay? Now there are some modifications to gram staining. Kopeloff and Berman's modification, Weigert's modification, Jensen modification, Preston and Morrell's modification. All these modifications are there in your book. You need to understand, you need to study. If asked in the exam, you need to tell, okay? Suppose this one is important, Kopeloff and Berman's modification. In case of the primary stain, we use crystal violet, but with this modification, we use methyl violet and as primary stain and basic fusion as secondary stain. Normally, in classic gram staining, we use crystal violet and safranin, okay? But in modification, we use methyl violet and basic fusion, okay? I am explaining you the first one, but the rest is self-explanatory, okay? Now we'll move on to the steps of gram staining. What is the first step? Smear is stained with para-rosaline dye for 1 minute, okay? You need to know the time for 1 minute. We'll use methyl violet here, crystal violet here. We pour crystal violet. As soon as we start pouring crystal violet, you will start your timer. Keep your watch. Everyone needs to have an analog watch during this practical. Pour the dye, wait for 1 minute, and then rinse with water, indirect water, okay? We have all shown you how to rinse the slide during the methylene blue staining. Use that technique. In your second step, you will pour grams iodine, keep it for 1 minute again, and rinse carefully. Okay? And then we'll move on to the third step. That is the most crucial step of gram staining. This will decide whether the slide you have made is proper or not. Okay? If you overpour the decolorizer, even gram-positive bacteria will expel the gram stain, okay, if you keep it for a longer time. So you only use acetone for 2 to 3 seconds. This is the most important step. You will use acetone for 2 to 3 seconds and then rinse it immediately. So you will not count like 1, 2, 3 in your watch. You will just pour the acetone. That will itself take 2 seconds and then rinse the slide, okay? And then on to the fourth step, that is the counterstaining. We'll use safranin, pour it, keep it for 30 seconds and then we'll let it dry and we will observe it under what objective lens? Oil immersion objective lens, okay? So under oil immersion, if it is gram positive, it will appear purple. If it is gram negative, it will appear pink. Okay? So let's go and do it for yourself.